seminar series link on the screen? Yeah, good. Thanks, Bob. You're the only one who can talk back to me today. Great. Well, good afternoon, everyone, and thank you for joining us today, and welcome to what is the second of our Institute of Ophthalmology seminar series. So this is a relatively new series which has been organised by the Deputy Theme Leads, so that's myself, Tessa Decker, Ryan McDonald and Adam Dubis. So we've invited a number of speakers from different areas of eye research with different expertise that lie within our particular themes. And so you'll have talks from researchers whose work you may be familiar with and is similar to you, or something that is completely different from what you're working on. So the hope is that this could inspire you to look at your research in different ways. So I would encourage you to make this part of a regular Wednesday afternoon slot to learn more about the wider aspects of research. And just to give you an overview, the seminar series is running on alternate Wednesdays to the Research Network series, which runs for an hour at 2:30. So there should always be something in your diary in the 2:30 slot. And for the seminar series, you do need to sign up for registration. So if you've made it here today, then you've obviously done that correctly. So well done. So before we start the seminar, I just wanted to go a bit through some housekeeping and how today's session is going to work. So we're delighted to have Bob Johnston with us here today. Bob is going to give his talk, so we'll all be with him for about an hour or so. And then we have half an hour for the early careers researchers. And this is to spend a bit of time with Bob and to ask questions. And these are more sort of wider discussions. So it could focus on the science, but Bob is also a great advocate for trainees and postdocs. So it could also be an opportunity to talk about, working in the US, career progression, defining a research area, or starting up a new lab. And then at 4 o'clock, we'd like to invite the PIs back for a more roundtable discussion, which is everyone's welcome to that. So like I say, during the session, your microphones and cameras are switched off. If you want to ask questions, the chat will be open. So you can either submit a question in the chat and we'll get to that at the end of the talk, or you can raise your hand and I'll open up the mic so that you can speak. So hopefully that's clear. So this just leaves me to introduce our speaker today. And we're absolutely delighted to welcome Bob Johnston with us. Bob is an associate professor of biology at Johns Hopkins University, where he studies mechanisms that underlie neural function during development. He was awarded his BS in biology from Drexel University in Philadelphia. And during his PhD studies with Oliver Hober in Columbia, he worked on neural specification in C. elegans and discovered, for example, new roles for microRNAs in neuronal fate decisions. And after this, he started working with Claude Desman in New York, where he published a number of high-impact papers examining the development and function of the visual system in Drosophila, with a particular focus on the photoreceptor cells. And Bob joined Johns Hopkins in 2013, where he's established his research using a combination of models, so Drosophila and human retinal organoids, to ask how retinal subtypes are specified. So this includes working on retinal ganglion cells and cone subtype specification. And this led to his recent science paper, which uncovers a role for thyroid hormone in specification in rods in retinal organoids. So with that, I'd like to hand over to Bob and thank him for joining us today. And I'm just going to stop sharing my screen and let Bob share his. So welcome, Bob.
Thank you so much. Hold on, let me get set up here. There we go. First off, I just want to thank Amanda for this awesome invite. It's really great and a little intimidating, especially when she's showing that schedule of really awesome speakers to be kind of sandwiched between Seth and Marla Feller. Or yeah, that's a little intimidating. So, or I guess it's an honor for me to be amongst those colleagues, especially my own colleague, Seth Blackshaw, who I'll be sharing some work that we've done with Seth actually as part of this talk. So what I'm going to tell you about today are how cone subtypes are specified in the human retina. And in particular, what I'm going to tell you about are how we use human retinal organoids to study this feature. And if you want to check us out online, you can follow me on Twitter, Bobby J. iGuy, or you can check out my website, or not my website, our lab's website, right there. And I just want to say that for everybody, but particularly for the trainees, if you have questions along the way, please, please, please feel free to raise your hand, drop a question in the chat. And maybe if I miss it, because I'm not used to this format of virtual talks, if Amanda could help me out and bring it to my attention if I miss it, I'd greatly appreciate it.
Yes.
So thank you so much. So I have been interested throughout my career in one major question that's been a major question in biology and neuroscience and retinal science for a long time, which is how do we get the different kinds of neurons in our body? And in particular, the retina has been an amazing model to interrogate this ever since the time of Cajal with his beautiful images, who hopefully you've seen, the multiple different kind of stainings that he did to identify the first neurons in the retina. And so we asked this question in two systems that are very similar and yet different. In the left, what you're looking at is the R7 photoreceptors of the fruit fly eye. And in the right, what you're seeing are the 3 cone photoreceptors of the human eye. And what you notice is some similarities and differences. One is that you've pretty much got exclusive fate. In this case, each of these is marked by the expression of a light detecting rhodopsin or opsin protein. In the left, we have expression of rhodopsin 3 here in blue and rhodopsin 4 in red. And in humans, you have expression of the red, green, and blue opsin. And so we take these two systems and we want to address a couple key questions. The first question we address is how do cells randomly choose between fates during development? We primarily ask this question in the context of the fly eye, because hopefully you can notice that pattern, there is no pattern in the fly eye, that each cell makes a random choice. And luckily during development, most of us are normally developing people, and as a result, it's very reproducible or stereotypical. But sometimes during development, there are random kind of coin flip processes that give you random patterns, and we use that system to address that question. Another big question we try to address in our lab is how are the cells in the human eye specified? This is a very big, broad question. We think there are tens, if not hundreds, of different subtypes of neurons, and we have started this kind of journey with these 3 cone subtypes. And finally, a new direction for our lab is we want to kind of build on what has been known and understood in model organisms to try to reconstitute the human visual system. We are in the early days of this, but our ultimate goal is to grow human retinal organoids and human brain organoids and connect them in a biologically and physiologically meaningful way, meaning not just put them next to each other and watch them connect, but rather trace the full axon outgrowth through the many paths to get from the eye to the brain. And today what I'm going to talk to you about is 3 stories dealing with how the cells in the human eye are specified, in particular these cone photoreceptors. So what do these cone photoreceptors do? Well, they are our main daytime and color vision photoreceptors. And what you can see, hopefully, for those people that are color vision normal, is this very beautiful kind of very dramatically colored rainbow balloon here. And people that have color vision defects, there are a couple main types. The first type is a loss of green vision, and you can have a slight, let me see if I get my cursor on here, I apologize. There we go. A slight loss or a complete loss. A complete loss would be called deuteranopia. If you lose, there we go. If you lose red vision, you would have protanopia. If you lose blue vision, what you'll notice is you see a very different kind of color vision. This is called tritanopia. And then finally, you could have a complete loss of green, red, and or blue vision, resulting in either monochromacy or partial monochromacy. So what I'm going to tell you is kind of the history leading up to our studies and how our understanding of these diseases led us to understand the genes. And then ultimately what we're doing now is try to understand the mechanisms of how we make these different subtypes. So again, these types on the left would be called red-green color blindness. Tritanopia is blue-yellow color blindness. And then you have things like monochromacy, which is total color blindness. So what are these cells that detect color? Well, the way hopefully most people in this audience understand is, light goes in through the lens, it goes all the way and hits the back part of your eye here in the retina. And so in the main part of the retina, we have different cell types. I'm sure that Seth went through a lot of those cell types last week, so I'm going to just focus on the study type we're going to focus on, which are these red, green, and blue cone photoreceptors. Okay, and just because sometimes I'll be switching back and forth, the kind of official name is blue or S for short wavelength detecting, green or M for middle wavelength detecting, and red or long wavelength detecting. Most of the time I'll be using blue, green, and red, but occasionally you'll see for certain reasons I'll call them S, M, or L, but I'll be sure to indicate the difference. Again, it's either blue, green, and red, or S, M, and L. All right, so what is the key kind of factor that determines how these cones work? Well, the key factor is something called an opsin. These are light detecting proteins, G protein coupled receptors, okay? So what are opsins? Again, these are opsins or G protein coupled receptors. They contain a light sensitive chromophore called retinal. And in short, I'm not going to go through the whole pathway today, but what I'm going to say is when light hits these, there is a shift and you get this triggers these opsins, and this triggers the pathway downstream. So, what makes these different opsins different from one another? And what makes it different is the sensitivity of these opsins. So you can see here the peak sensitivity of the blue opsin is different from the green and red. And noticeably, the blue opsin is very different from the green and red, whereas the red and green opsin are more similar. And you're going to see this is kind of mimicked in terms of their gene regulation and also even in their placement in the genome. So in terms of, and also, by the way, in their protein structure. So we can see here, if we compare the M and S opsin, the green and blue opsin, in black, we see amino acids that are different, and in white, we see amino acids that are the same. And what you can see is there are some amino acids that are the same, but there's a lot of differences between the M and S opsins. But now if we compare the L to the M opsin, what we notice is they are super similar. They're almost every amino acid is the same, and there's only a handful of amino acids that actually make these different. And what I'll discuss later is this has made this very challenging to study as a question is how would you express either the L or M opsin because of that similarity. So the next question that's important to know is where are these opsin genes in the genome? And I just want to say I don't have that a lot of this work was originally done by Jeremy Nathans, another colleague here at Johns Hopkins. And what he found was that the S opsin gene or blue opsin gene has this very simple structure where you have a simple enhancer connected to this S or blue opsin gene. Now for the red-green, it's a bit more complicated. For the red and green, they're actually in tandem. So you have the red gene and the green gene, and they are controlled by a single enhancer called an LCR or locus control region enhancer. Okay? And so again, notice the similarities. We have the blue kind of its own little special place, and the red and green are inherently kind of linked. And you'll see as I go through this talk again, how these decisions pan out in terms of the development and specification of these different cone cells. So, what do we know about these opsin regulation? Because this is where we're going to start our journey and try to figure it out. Well, when we entered the field, there wasn't really that much known. And what we did know is there seemed to be two key steps. And it kind of follows the layout of these genes. The first step is there's this decision to either be to express the blue opsin to the exclusion of the red-green opsin. So in other words, we're saying, okay, we're going to express this blue gene. Okay, great. We're not going to express the other one. Or we're going to choose this other path where we're going to choose either the red or green fate. Okay. And throughout my talk, I'll call this the blue versus red-green choice. Okay. In the second step, there's a second choice where this enhancer is thought to loop to either the red gene or the green gene to dictate that final choice. So again, the first choice is to be either blue or red-green. In the second decision, if you take the red-green path, you then make a second choice to be either red or green. So all right, so this kind of has provided the background. And when we started this work in about 2014, this is where we started. And again, I just want to say that this was work that was really inspired by our fly work, and I just want to again give lots of props to all the work in model organisms, because it's really set the foundation for now what we can do and try to understand in human systems. So the outline of the talk today, what I'm going to tell you about is the choice between blue versus green-red cone photoreceptors, how the fovea, or the specialized high acuity region of the retina, is specified, and the green versus red choice. And so these are works by 3 different grad students, and it's their main thesis work, so I'll describe each of these. So the first project is by Chiara Eldred. This was published in 2018, and she was, a real superstar who helped establish this system in my lab. So walking into this, I've kind of described the basics. And as developmental biologists, one of the first questions we wondered is, well, when are these cone subtypes generated? And we went through the great work of several scientists, and one real standout was the work of a ophthalmologist, or an eye scientist, Anita Hendrickson. And what she had done throughout the years was she took a look at expression of opsins, or the generation of these blue versus green-red cones in fetal tissue. And very simply, to summarize this graph, the simple relationship is that the blue cones were made first, and they kind of leveled off, and then the green-red cones were made later. Okay, so the simple relationship seemed to be temporal: blue first, green-red later. And so we wondered, okay, could we figure out anything about this temporal mechanism? And so with this hypothesis that blue cones became before green or red cones. And the big question is, well, how can we study cone specification in humans? I often joke, we just had our grad student interview weekend, and I often joke to people, it's like, how much do you want to get into Hopkins? Because we definitely could use donor tissue. Which one would you donate? But some people get that joke, some people don't like that joke as much. But bottom line is this: it's hard to get human tissue to study, and there's a lot of ethical and other concerns. So I was really inspired when I saw a talk in 2012, actually, by a really phenomenal scientist who has since passed away, Yoshiki Sasai, who was one of the first to really develop these human retinal organoid systems. And when I joined Johns Hopkins, my lab was exclusively using the fruit fly model system. And I had given a talk, and I was very fortunate to run into 2 colleagues, Don Zach at the med school, and a postdoc at the time, Carl Whalen, who now has his own lab at UCSD. And I'm really grateful to these folks who helped me get this system up and running in my lab. And now we use it as one of our main models to study neuronal cell fate specification. So what do? We want to grow human retinal organoids in a dish. So what we start with are human either iPS cells or ES cells. We do what's called a force aggregation, which is just the fanciest word ever for letting the cells fall to the bottom of the tube. We then add a series of signaling agonists and antagonists. And what you're seeing here is primitive neural tissue starting to grow from those stem cells. By day 8, what you're now seeing is the beginning of differentiation of primitive retinal tissue. We then dissect these retinas off so that each one is now growing in the dish on its own. And it's, it doesn't look so great. It's kind of amorphous still. But by day 43, you see one of my favorite pictures, which is this still primitive, but very clearly retinal tissue, where you have the different nuclear layers of the retina starting to differentiate, and this kind of curvature of the retina. So when we came into this process, we thought, all right, we've grown organoids. Step 1 done. But we're again kind of naive, because we didn't really totally think it out. And what we realized is it takes a long time to grow human retinas. And to get at this, Chiara really had to push the system beyond what had been done previously. So she grew these organoids out past day 43, out to day 281, day 329, and beyond. And these are really challenging experiments. We do these in antibiotic-free conditions. And we were kind of worried, is this fuzz on the outside? Is this some kind of contamination? What is this? But in short time, we realized this is actually a good thing. What you're seeing here is actually the outer segments of the photoreceptors growing out from the outer nuclear layer. And we were very excited to see this. And we were even more excited when we then stained for the opsins with immunohistochemistry. And what we saw here are the blue cones in blue and the red-green cones in green here. And throughout this part of my talk, I'm going to call them again the blue cones and the red-green cones, Because if you remember, the red-green cones are so similar that we don't actually have an antibody that can discriminate between them. So we use antibody staining for this part. And again, we are going to be talking about the blue cones marked with the S opsin antibody and the red-green cones marked with the LM opsin antibody. All right. So great. So now we have our hypothesis is that blue is before red-green, that we see that data from the human tissue, and we have these organoids. So the next big question is, can we even use these organoids? We're developmental biologists. Just because they make the cells doesn't mean they make it in a way that's similar to actual human biology. So what Chiara did is she asked, are blue cones generated before red-green cones in organoids? And indeed they are. So what she saw at day 150 is exclusively blue cones. Day 190, these blue cones the number of blue cones kind of remains stable, and now we start to see red-green cones. And then by day 290, we see a significant increase in the number of red-green cones. I want to emphasize that these are all fixed samples, so these are actually different organoids, but we'll see in a second as the numbers are fairly consistent. So when we quantify this with time over days on the x-axis, you can see we took this out to a full year. And the cone density here, again, what we see is that over time we have no blue or red-green cones, and then we see blue cones only, which pretty much stabilizes. And then we see a steady increase in the red-green cones, suggesting that indeed this recapitulates human biology. And so the next question, of course, is, well, great. So this does this. Well, now we want to get mechanism, right? So what are we going to do? So to get at this, we now turn to the great amount of information provided by model organisms. And what's interesting is that model organisms, the cones, have very different patterns. What you're seeing here is a mouse eye, and we can see is green cones, co-expressing cones, and blue cones. And then what I'm going to show you next is the fish eye, which actually has 4 different cone subtypes, and they're in this perfect grid. So it's very different from the human eye. But what ties it all together is a receptor called thyroid hormone receptor beta. And what several groups, including the Forest group and one group, showed was that this is a key to controlling cone subtype specification. So with this said, we came up with this hypothesis that this thyroid hormone receptor beta drove red-green fate and repressed blue fate. And so to test this, Chiara used CRISPR to generate knockouts of this receptor with the hypothesis that we would see only blue cones. And so now Chiara did this experiment. I want to emphasize that all these experiments, because organoids do have variability and heterogeneity, we always do them with internal time-matched controls. And what she saw, again, is in our controls, we get a mix of blue and red-green cells. And then we saw this really beautiful result that in the thyroid hormone receptor beta null mutant, we see a complete conversion that we get retinas with only blue cones. And this was important on a couple fronts. One, from a mechanistic point, it was important. Like, great, we identified a key gene. But I think more importantly for my own group and for the organoid field in general, is this showed us that we can really study developmental biology using these human retinal organoids. They are far from perfect. I want to just emphasize that. When you read these in the papers, people emphasize how similar to retinas they are, but they're not perfect, but they can serve as really excellent models to study these questions of human biology. So awesome. So now, of course, great. So we have a receptor, we have a gene. But of course, the next question, well, what regulates thyroid hormone receptor beta? And thyroid hormone receptor beta is a receptor for thyroid hormone, and the active version of thyroid hormone is called T3. And so going back to our timeline, we thought, okay, how could this be working? Well, if thyroid hormone receptor beta promoted the red-green fate, then that would suggest that T3 is acting late in development. So we wondered what would happen if we now added T3 early and throughout development. Would we see some kind of effect? So Chiara did this set of experiments. Again, in controls, we see a mix of these blue and red-green cones. And again, we see a very dramatic effect when we add thyroid hormone throughout development, generating retinas with almost complete red-green fate. So awesome. So now we have T3 thyroid hormone activating this thyroid hormone receptor to promote red-green cones and to suppress blue cones. So of course, the next question is, well, what regulates T3? We want to move up this pathway. But at this point, you might be thinking, okay, Bob, you're talking a lot about thyroid hormone, but there's kind of a big problem, right? What regulates T3? Well, we're organoids. There is no thyroid gland in the dish. So what this told us is that somehow the retina itself must be regulating this. And this is actually commonly known that there are enzymes in specific tissues that can modulate the levels of thyroid hormone. So we next sought to kind of address this by looking for different regulators of T3 thyroid hormone levels. So again, what we said is, okay, we have a hypothesis that late, there should be high T3 to generate these red-green cones, and then early, there should be low T3. How can we do this? Are there enzymes that activate or degrade? What's going on? So to get at this question, Chiara teamed up with another grad student in the lab, Sarah Hadniak, and conducted bulk RNA-seq at multiple time points across a full year of organoid development. And what we're looking for is these key regulators. And I'm going to quickly kind of dip into some of these players, but don't get too bogged down. Just think about whether they're activating or degrading thyroid hormone. So T3 is coming from an inactive form called T4, and both of these forms are degraded by an enzyme called deiodinase 3 or DIO3. And so our prediction is early we would have low thyroid hormone, and we predicted as a result, perhaps we would see high DIO3 or deiodinase 3 expression. And indeed, this is what we saw. When we looked at our bulk RNA-seq, we see high expression levels of DIO3, which decrease over time, consistent with the idea that DIO3 degrades thyroid hormone to allow for the generation of blue cones. Great. So this could be doing a job on its own, but perhaps there are other parts of this pathway that play a role. So T4 is converted to T3 by an enzyme called deiodinase 2 or DIO2. And our prediction here is that late we want high T3 levels, so perhaps deiodinase 2 is turning on over time. And indeed, this is what we saw. Over time, DIO2 is increasing, consistent with the generation of the red-green cones. And consistent with the idea that DIO2 would be converting T4 to T3. So this took us to a model where we see potential temporal regulation of thyroid hormone signaling to generate these two different cone subtypes. Early, we have expression of DIO3. This is going to degrade thyroid hormone. As time goes on, we're going to generate naive cones. They're in an environment that'll allow for the default kind of blue cone fate. As time moves on, this DIO3 is going to turn off, allowing for T4 to exist. DIO2 is going to turn on and convert that T4 to T3. When new cones are born, they're going to have high levels of thyroid hormone signaling, and they will generate the red-green fate. So together, for this first part of my talk, what this told us is that over time, thyroid hormone signaling is increasing, generating the late-born green-red cone fates. And so what I'm going to tell you next is that there's another signaling pathway, retinoic acid, that plays a role early in cone subtype specification. But before I move on to this question, and the big question I'll be addressing are how are cones patterned in the human retina? But before I move on to the second part, if anybody has any questions, fire away. I'll take questions before I move on to the second part.
Hi, we've got one question from Matteo.
Okay, great. I can't see the chat or anything, so just fire away. Don't be shy.
Matteo?
Hi. Yeah, can you hear me? Yeah.
Yes.
I was wondering, because you've been talking in terms of S fate and LM fate, I'm wondering how much you think this could be S-opsin expression or LM-opsin expression and something related. I guess, like, you probably agree, like, I guess early in development, even LM cones express S-opsin, and then these get switched to LM, right?
So yeah, so this has been actually, this is a great question. So let me rephrase the question, make sure I get it. The question is, first off, do the S cones convert into ML? And because the way that I proposed it is that it maybe wasn't totally clear, so I'll clarify it a bit. What we initially thought was that you generated S cones, and there was kind of a time point where new cones were generated, and they would all be LM cones. What we think now is there actually is some conversion of those blue cones into red-green cones. And actually, the next part of my talk, I'll be covering that.
I see.
Yeah.
Cool.
Does that address your question?
I think so, yeah. So I guess what I was thinking is whether when you suppress by adding T3, when you make almost all of them into LM, I was wondering whether the ones that are remaining that are basically are resisting these T3 are the, let's call them truest cones, and then the others, and yeah, I guess you're nodding, so maybe, yeah.
Yeah, that's a great question. So we don't know those. What I'm going to talk, again, this in the next part of my talk, where I'm going to show you some single-cell RNA-seq data where we kind of get at that, but it still doesn't address those kind of residual blue cones that seem kind of not susceptible to that signaling. We're not sure yet. What I can tell you is that in another project, Christina McNerney, a grad student in my lab, has been playing around with the DIO3 and DIO2 enzymes. And what she sees is that when she knocks out, when she actually makes a hypomorph of DIO3, we kind of get a counterintuitive result, which when it takes a little bit, together with some other data, it makes sense, is we actually see more blue cones. So DIO3 degrades it, the hormone, and you would think that by knocking that out, you would get more T3 and more red-green. But what we found is that T3 also plays a secondary role in actually generating the cone cells. So if you knock out major players in that, you kind of perturb that. And what we've also found is there's feedback. So in other words, if you knock out or just kind of tune down the function of one of these regulators, it changes the feedback of those mechanisms. So bottom line is, sorry, that was a very long-winded way to say that what we're seeing now is that there seems to be plasticity, and I'm going to show you that in a minute, and that also there's feedbacks, and I'm not going to be talking about that in this talk.
Okay, thanks.
Thank you.
Bob, I've just got a quick question, a basic question. Yeah. You're obviously showing that there's regulation of the thyroid hormone by the retina, but where's the thyroid hormone coming from?
Yeah, that's a great question. So we think, I mean, the only place it can come from is the media, and we have tried using mass spec to quantify that, but it's on the femtomolar scale. So it's been really challenging. We're actually revisiting it. Before the pandemic, my grad student, Christina, again, who I mentioned, was working to do some biochemical approaches. But when the pandemic hit, my lab is not really a biochemistry lab, and it was new to us. So we kind of put that on hold. We're now going back to revisit that kind of on two scales. One is, can we detect the inherent ability of the organoids to change T3 and T4 levels? And then generally the media and the organoids, what is the absolute levels? I think we'll probably find more in terms of assessing the enzymatic activities, because the actual concentrations are very, very minuscule.
Okay, great, thanks.
Thank you. All right, so if there's no other questions on this part, and again, don't be shy. If you guys have questions, if you folks have questions along the way, feel free to interrupt me. All right, so the next part of my talk, so the first part was generally blue versus red-green. The next part, we're really gonna kind of chisel down and focus on a specific region of the retina, and how can we use organoids to kind of make hypothesis and test those hypotheses about how these specific regions of the retina could be generated. So this is work by Kasia Hussey, and I gotta say, she really spearheaded this. She came into the lab, we do rotations here in our grad program, and since her rotation, she was like, I wanna do this. I don't know why she got so into it, but I love that spirit, and it really pushed this project. So as I mentioned at the beginning of my talk, light goes in through the lens into the eye, hits the retina, and I talked about kind of the macula, which is a cone-rich region, but it has a mix of blue and red-green cones. There's another region called the fovea, which is a very specific, unique region, because first off, there's this kind of pit where you have only red and green cones, okay? And this seemed to be a perfect question to fit to tie to the first question is, it seemed like the decision point is this blue versus red-green. And in particular, in this fovea region, there must be mechanisms that really push this system to only generate these red-green cones. And so, and one key feature of this is a very important region medically, because this is a key part, the center of the macula, which enables high acuity vision. And as I already mentioned, just to reiterate, the fovea only has red-green cones. And so let me tell you a little bit about the layout of this region. If we kind of rotate the eye and now look from the other side, again, if we zoom in on this kind of the broader region would be the macula, what we see is actually outside of this called the periphery, we see mostly rods in gray, and then our mix in of blue, red, and green cones. As we move further in, we have in the macular region, we see an increase in the overall cone density and less rods. And then as we get even further into the fovea, this density increases further. But finally, as we enter what's the very center, the foveola, we see, or the center of the fovea, we only see red and green cones. So what we wanted to address specifically is how can we prevent the generation of blue cones and generate only red-green cones? And again, as I've been saying, this kind of gets at this question of the first choice between blue and red-green cones. And the big picture question that Kasia wanted to address is how is the fovea patterned during development? And so to get at this, she kind of revisited and did herself some of the experiments originally done by Anita Hendrickson, looking at different regions of the retina in fetal development. And so this is a very large chunk of retina, but let me zoom in here. What she saw at 10-week retina was very specific, that within this fovea, there were blue cones. And this was a little counterintuitive because you would think a region that would eventually not have blue cones should start with no blue cones. But Kasia saw this, and this was also seen originally by Anita Hendrickson. So we were happy to see that we recapitulated Anita's initial findings. But then interestingly, by day 14, we see 3 different kinds of regions. In this central region, the future fovea, what Kasia saw now was green exclusive cells, which is kind of what we predict to see. In other words, at that point, the blue cones are already gone, and we only see red-green cones. But interestingly, in the continued developing part of the fovea, so this is still part of the fovea, but a little further out from the center, we saw multiple things. We saw red-green only, but then we also see co-expressors that express both the red-green opsin and the blue opsin. And so this was a little confounding, that there were some reports of this, but this really suggested to us that maybe there's a plasticity between those. And then since development starts in the center and moves out, and blue is before red-green, in this outermost region during development, we saw only blue cones, consistent with the retina developing from the inside out. Now, unfortunately, due to limitations on accessibility of fetal tissue, we can't look further out, but this already gave us several clues to how this could be working. So what do we see in this? What do we see in this fovea? So first, what we think we see is that there's less blue cones initially, and though those blue cones are not present later. Okay, so that's the 2 kind of things or ideas that we want to test using these retinal organoids. And first, let me talk about how there could be blue cones less initially. And so as part of this, again, the big question is what controls this cone patterning in the fovea? So what we did is we went to several reports, some done by in situ hybridization, some done by a single cell RNA-seq, and we really scoured the literature. And what Kasia came up with, it was a very nice hypothesis that retinoic acid could be playing a role in this decision. And the rationale is that consistently what she saw across multiple groups is that in the fovea region, there's expression of enzymes that degrade retinoic acid, whereas in the periphery, there's ones that synthesize retinoic acid. And so those enzymes that synthesize are called ALDs, or ALDH1A1 and 1A3. And then for the sake of today's talk, we're going to focus in on this CYP26A1 that's expressed at high levels in this fovea. And interestingly, what it does is it degrades retinoic acid. So from this, Kasia had the hypothesis that low retinoic acid promotes red-green fates, because keep in mind, in this fovea, we only have red-green fates. Okay, so we want to limit the number of blue. And so what that would say is that in the periphery, normally, we might have high retinoic acid to promote blue and repress red-green, whereas in the fovea, we'd have low retinoic acid, which would prevent the generation of blue cones, but allow the generation of red-green cones. So to address the first part of this hypothesis, Kasia just wanted to say, okay, well, does retinoic acid influence cone fates using organoids? And so what she did is she looked at several different concentrations. Indeed, what she saw is there was regulation. So at low RA concentrations, we see retinas that are very red-green rich. We see very few blue cones. As she increases the levels to a medium level, she sees an even mix. And then when she goes to high levels of retinoic acid, she sees very blue cone-rich retinas. And so when we quantify this, what you can see is this very nice relationship where at no RA, we see significantly more red-green cones, And then as we increase the levels of retinoic acid, we change the ratio so there's much more blue than red-green cones. All right, so that's great. That's pharmacological evidence that it plays a role. But what she really wanted to do was say, okay, let's get to the molecules here. And what regulates retinoic acid? So again, CYP26A1 is our primary candidate. And the hypothesis here is that CYP26A1 would repress retinoic acid, allowing more red-green cones. But if we knocked it out, perhaps we would enable an environment that would have higher retinoic acid, enabling the generation of more blue cones. So Kasia tested this hypothesis using CRISPR to knock out CYP26A1, and she asked the question, how does CYP26A1 influence cone fates in organoids? So what she saw now is in our controls, she saw very red-green rich organoids, and in the knockouts, we see a dramatic increase in the number of S or blue cones in those mutants. And here's just the quantification. Again, in these conditions, in our controls, we see very red-green rich organoids, and in the knockout, we see an upregulation of these blue cones. So together, what this suggested is that CYP26A1 acts in the fovea to lower retinoic acid to allow for more red-green cones. So, all right, so we've kind of addressed the first part. CYP26A1 lowers retinoic acid to promote red-green cones and suppress blue cones initially. But now what about the second part? That these blue cones are not present later. Because we know in adults, we've done this ourselves, and many other groups have seen this through many different ways, is that that central region is red-green only. So how are we going to get rid of these blue cones? Well, the initial hypothesis from the Provis group was that these blue cones migrated away. And this was a while ago, and they didn't have any direct evidence. It was more just based on the idea that they didn't see cell death and they didn't see the blue cones later, so they must have moved away. What I'm going to suggest is an alternative model, which is that these blue cones actually convert fate. And this kind of gets to the question I was asked. I'm sorry, I forgot your name. Had a very good question during the last section about this system. So, we now return and said, what else regulates cones in the fovea? And so we went through this list. This is from Yi et al., very nice single-cell RNA-seq work. And we said, okay, what is consistently high in the fovea? And we noticed one of our friends here, DIO2 or deionase 2. And if you remember, this is a regulator of thyroid hormone. And DIO2 upregulates T3. And DIO2 is highly expressed in the fovea. And this is expressed even into adulthood. So what it says is, for whatever reason, the fovea has high DIO2 throughout its lifetime. And with this, we kind of came up with this hypothesis that sustained DIO2 and hence T3 levels affects cone fates. And our prediction is that then that high T3 or sustained T3 would promote the generation of these red-green cones. So the next question we said is, well, how does the timing of T3 signaling affect cone fate? And to get at this, we're going to take a step back to our timeline here. And what I showed you already is that we get blue cones that are maintained throughout development, and then red-green cones are generated a bit later. In our controls, we see a mix again of blue and red-green cones. And the experiments I already showed you is that if we have T3 throughout time, we see almost exclusively red-green cone-rich organoids. So now we wonder what would happen if we played with this timeline. So what Chiara did was do these experiments at different time points, at key time points. So when she shifted it now to the time point where blue cones are generated, what she sees is basically the same thing. Okay, well, maybe you're still converting those blue cones before they're even made, and no big deal. But then what she did is after the time point of blue cone generation, when she adds T3 for a long time, she still sees these red-green cones, suggesting that they must be either lost or converted. And to directly address this, she thought, okay, well, what happens if I add it very late for a short amount of time? Maybe I can catch these cells in the act. And indeed, first I'll say that what this suggests is that you really need prolonged T3 to generate these red-green cones. And then she wondered, okay, what if I just do it for 10 days? At this point, the system is more or less set up. Those blue cones are stable. When she did this, interestingly, what she saw was she kind of caught these cells in the act because she now saw co-expression of blue and red-green opsin in these cells, suggesting that if we upregulate thyroid hormone levels late in development, we can actually convert the blue cones to the red-green fade. So the next question we have, of course, okay, we have this hypothesis that the blue cones are converting into red-green fade. What can we do to address those? So what Chiara teaming up with Kasha did was we said, okay, let's go to single-cell RNA-seq and see if we can see changes in the cells across the cell fates. I've been talking pretty much exclusively about opsin expression, but there are other genes that define the fate. So let's take a look. So what they did was they took our control organoids, our pulse organoids, and our sustained T3 organoids and said, okay, what would we say? So we did single-cell RNA-seq. You've hopefully seen these a lot, these plots. We're going to focus in on these cone cells here. And what they wanted to do is say, okay, how are blue associated genes, in other words, genes that are normally highly expressed in blue cones, affected when we add T3? So in our control, we're going to see expression of these two genes. In our pulse, the expression of these genes starts to decrease. And then if we have sustained expression, we see a dramatic decrease. So what this says is that in the blue cones themselves, the blue associated genes are decreasing. Now we ask the opposite question. What happens to green associated genes in the blue cones? So in this case, we see some expression in the controls. When we pulse, we see even an increase. And then we see a more dramatic increase if we sustain the T3 throughout time, supporting the idea that indeed these blue cones are converted into red-green fade. And so together what this supports is that prolonged T3 converts blue cones into red-green cones. And so together, this supports a model in which in the fovea region, we have naive cones that are generated. CYP26A1 is expressed to limit the levels of RA. So we're going to limit the number of blue cones that are generated. Over time, we're going to crank up our T3 levels. And as new naive cones are made, they're going to take on the red-green fade. But interestingly, the blue cones are going to start to convert to the red-green fade. And the net effect is by adulthood is that we have a fovea that is red-green rich. So together, tying both parts of the talk so far together, what we see is this temporal roles of two key signaling pathways. Early retinoic acid seems to promote early fates like blue cone fate, and later thyroid hormone promotes these green and red cone fates. So before I get to the next phase, so the next question I'm going to address is, well, I've been talking a lot about the blue versus red-green. The next question, the third part of my talk that I'll address is this green versus red cone fate. But before I get there, does anybody have any questions?
We have a couple of questions. Let me just open the mic. Anna? Anna, have you a question?
Thank you. Yes. Hi. Thank you, Robert. So I wanted to ask you, you've done T3 addition at different time points, and I wonder if you've done the same with the retinoic acid concentrations that you've shown?
Yeah, that's a great question. So we have done a bit of that. We only see effects early. So in other words, it's kind of the opposite of the T3. When the T3, when we add it late, we can convert it late. But with the retinoic acid, it seems like only the early time points matter. There is a little bit of a challenge there because with the retinoic acid, and actually we'll talk about this retinoic acid thing a little bit more in the next section. But with the retinoic acid, we cannot sustain high levels throughout development. So there's some challenges from a technical perspective. But overall, what we see is that retinoic acid early can make changes, but late cannot.
Okay, thank you so much.
Thank you.
And then we've got a question from Mike, if I can unmute him.
Thanks, Amanda.
Okay.
Thanks, Bob. It's great so far. I just wanted to ask, I noticed when you had your cells which were positive for both LM and S opsin in the fovea, the shape of the staining pattern seems to be different between the S and the LM antibody. Do you think that's telling us anything, or is that?
Yeah, that's a really great question. So what we do know is that, and I'm sorry, I can't even see who you are. So I don't know if you're an expert or like an undergrad who just started or whatever. So I'm just going to speak to you as if you're new to this. And I apologize if you're a super expert. So
I do know a bit about options, yeah.
Okay, great. So the cone cells have different morphologies, right? So the blue cones have kind of short and stubby outer segments, whereas the red-green cones have these long, thin outer segments. And also the localization of the opsins itself are different. The blue cone opsin tends to go through the entire membrane, whereas the red-green opsin is localized to the inner and outer segments. So the different localization is one aspect of it, but also we think that there's a different temporality. So in other words, the blue opsin is generated first, so it looks more peripheral, like basically at the membrane, the cell membrane, and the red-green opsin seems to be more central. And what we think is happening is it's just being newly produced and hasn't had a chance yet to go out to the cell membrane.
Okay, thanks.
Thank you.
Okay, I think that's it for questions now.
All right, great. And I know I'm running a little bit late. This last section is a little bit shorter than the others, but thank you to everyone for hanging in. And I hope that this last part is very exciting, I think, and I hope that you hang in. If you don't, that's cool, but I hope you hang in. All right. So this last section is really, the fovea was getting us to kind of very human-specific questions, but we still had a lot of fantastic work in model organisms that really gave us hints. This last question is very human-specific, and that made it particularly challenging. So this is work from Sarah Hadniak, a recent graduate from the lab who's now figuring out where she's going to do her postdoc. So the challenges in particular are a couple. So first is it's very poorly understood. Again, this is specific to primates amongst mammals. And because, you know, if you have pets, you likely know that your cat or dog is quote unquote colorblind. They're not actually colorblind, they're dichromats. They have blue and green opsins, but essentially they're not trichromatic like we are. The other challenge is that the red and green opsins are extremely similar. They are above 90% identical on the RNA level. So even doing RNA in situ hybridizations was not possible previously. So these are just some of the challenges within this. And as I already mentioned, this is unique to humans and old world monkeys. And again, there just hasn't been a great model. So we thought, okay, let's use these organoids, because that's what we want to do, is take these human retinal organoids to study aspects of human biology. So entering this, there are kind of two non-exclusive models, and we wanted to kind of see where we could fit in here. And so there's a stochastic model, and this was originally proposed by Jeremy Nathans, again, a colleague here at Hopkins. And this was based on an idea that these red and green opsins are close by on the same chromosome. And there was data using transgenics in mice suggesting that perhaps this LCR could loop to the red opsin to give you red fate, or loop to the green opsin to give you green fate. Another model was a spatiotemporal model based on physiology and some gene expression studies. And what this simply stated is that the late-born peripheral part of the retina is very L-cone rich. And this was suggested by microspectrophotometry, multifocal ERGs, and qPCR. And together, but one issue here is there is no direct visualization. And so the very first question we wanted to address is how are these L and M cones distributed? And this is a really big challenge. Again, these are over 90% identical on the RNA level. So what Sarah first had to do was develop an in situ approach, taking advantage of single SNPs in these RNAs. And this is with a company that has this technology, but they do not even have a fluorescent version yet. So the experiments I'm going to show you are colorimetric. And what she was able to do was develop this in situ approach. So indeed, we could distinguish between the red opsin here and the M or green opsin here in this bluish kind of green. So using this assay, what Sarah did was take a look across several retinas at different regions. And what she saw, and it's still in the works of doing to get her kind of numbers of different human eyes to study, is that indeed in the periphery, it's extremely red or L-cone rich. Even in the mid region, it's still red or L-cone rich. And then in the central region, now you see your mix of M and L cones. So this supported the idea that perhaps spatiotemporal mechanisms played a role in this process. So the next question we wondered is, well, how are M or green and L or red opsin expressed during the development? Again, these are challenging experiments because we have to go to kind of RNA-seq data that's available on fetal tissue. And in short, what we saw is that over time, so the x-axis here is different time points during fetal development, what we saw is that the green opsin will turn on, and in adults, we see expression of the green opsin. Interestingly, what we saw is essentially no expression of this red opsin in the developing fetus, but we did indeed see it in the adult retina. So what this suggested is a hypothesis that indeed there was some temporal aspect to this process, that indeed you would get blue cones, then green cones, and then red cones. And so, of course, great. Okay, so we have some observations here about the spatiotemporal layout and dynamics of the system. But the next question is, well, what specifies this? We really wanted to get into some aspect of the mechanism. And we took a step back and said, okay, well, thyroid hormone and retinoic acid play key roles otherwise. Could they be playing key roles here? And what I'm not going to show you is that we tested thyroid hormone. We saw no apparent roles for that. So now we turn to this retinoic acid here and said, okay, could retinoic acid be playing a role? In particular, we think it's promoting earlier fate. So could it promote this early green cone fate? And so the next question Sarah addressed is, how are M or green opsins and L or red opsins expressed in organoids? And what she found is that they are very red cone rich, kind of similar to what we see in the human retina. We see mostly red cones here, with a mix of some green cones. And again, what she wanted to say is, okay, we have this hypothesis that early retinoic acid promotes green cone fate. And so to address this, what she said is, okay, let's look at adding retinoic acid and then getting at time points. And this kind of goes back to the question that was asked earlier. But in this case, you'll see how this plays out. So during our differentiation protocol, we said that day 43 is when the organoid is basically generated. Cones are born between day 60 and day 80. Day 130 is when the opsin first turns on. And then usually we assess kind of terminal differentiation at about day 200. So in our normal conditions, we see a mix, mostly red cones, with a spattering of M or green cones here. And she wondered what happens when you add early RA. Indeed, what she saw was a dramatic effect where we now see a dramatic upregulation of the green or M cones and a suppression of the red or L cones. And again, to get at the question that was asked before is, does RA specify green or M fate in these undifferentiated cones? Because a big kind of topic in the field is it a gene regulation choice at the time of opsin expression, or is it really a developmental process? And what we thought is, well, if we do it before the cones are even differentiated, that this would be an argument for that timing. So what Sarah did then was basically extend this RA simply to day 60 or day 80, and she saw the same effect. In essence, at both of these time points, we see a dramatic upregulation in the population of green cones and a suppression of the red cones, supporting the idea that retinoic acid can act early to promote the green fate and suppress the red fate. Now, again, getting to the question that was asked, very prescient question, does retinoic acid convert fates? I talked about T3 or thyroid hormone converting fates. What about retinoic acid? And so Sarah did this experiment adding retinoic acid late. And what she saw was that retinoic acid was not able to convert fates late. And this was important to us for two reasons. One is it said, okay, this is going to be different than the thyroid hormone relationship. But also what it said is you can't go backwards. Retinoic acid promotes early fates. If you add it late, it cannot go backwards. So what I've shown you here is that early retinoic acid promotes this green fate. But what other evidence would support this going back to real human tissue and humans in general? So to get at this, we next looked at the temporality of retinoic acid regulators. And what we saw is that key receptors like NR2F2 is pretty evenly expressed. Okay, it doesn't seem like the receptor is playing a key role. CYP26 genes that I mentioned earlier seem to be pretty evenly expressed. So that seems that it doesn't play a temporal role in this process. But what you saw looking at ALD, which are retinoic acid synthesis genes, is that they decrease over time. They start high and turn off over time, consistent with the idea that we want early retinoic acid early in cone specification and decreasing over time. We look for other clues. So we look for clues in actual humans. And to get at this, we took advantage of the fact that there's a huge amount of variation between the red and green cone fates. So in normal humans that have normal color vision, you can see ratios of 3 green to 1 red, 1 to 1, or even out to 1 to 17. So it's dramatic variation, and we thought this is a great quantitative trait that perhaps we can get an idea of how this works. We collaborated with the Jay Knight's group and a great collaborator of mine, computational collaborator, James Taylor. And what we saw is that across this population, we see pretty much a nice normal distribution. If we look at percent red cone fate, there's some technical normalization errors. That's why you see a slight numbers above 100%. And what we did is for over 700 males, we took their DNA. We limited this to males specifically because this is X-linked. So we thought we would expose variation by looking at X-linked, by looking at males exclusively. And then we gathered DNA and we sequenced it. And particularly because we were looking for non-coding changes, we sequenced the red-green locus plus 20 candidate genes, including regulators of the thyroid hormone pathway and retinoic acid pathway. And we do what's called a Manhattan plot, where the y-axis is the correlation of a particular SNP with a phenotype. What we saw was not so great. So for most of these genes, we don't see a relationship. Part of this could be due to our very small sample size. Typically, you want to do this in the thousands of different. We only had hundreds. But what we were very excited to see was that indeed we had at least one significant SNP, and this was in NR2F2, the receptor for retinoic acid. So again, consistent with the idea that retinoic acid is playing a role in this process. And again, just to say again, is that the cone ratio is associated with the variant NR2F2, and that NR2F2 is a nuclear receptor that mediates retinoic acid signaling. So to summarize this last part before I go to my final conclusions, the green and red cones specification is a regulated process. There may still be stochastic features to it, but we can actually regulate it in the dish. Green cones are generated before red cones. And finally, this early retinoic acid specifies green cones in organoids. So kind of broader conclusions, what I've shown you is that retinoic acid plays a key role early. These regulators, the interaction of these regulators is such that retinoic acid signaling will decrease over time. Thyroid hormone increases over time to generate the late-born cells. And overall, what I've shown you is that organoids are a great system that recapitulate human retinal development, that thyroid hormone and retinoic acid signaling specify human cone subtypes. And what we really feel is that our work is advancing organoids as a model to study mechanisms of human development. And in the final couple seconds, I'll just say that there's many outstanding questions and ongoing work that we're working on, including the next big question. Well, what controls thyroid hormone and retinoic acid signaling? And so what Christina McNerney is working on is studying how DIO3 and DIO2 work. And what she's finding is a key kind of quorum sensing mechanism where the number of cells expressing each of these kind of ultimately controls the fate choice. We're also studying how this system evolved, particularly in primates. And to do this, what Joanna Hagen is doing is developing organoids from multiple different non-human primate species, including macaque here. Here is the organoid. You can see the red, green, and blue cones, in addition to DAPI here. She's done this for macaque and marmosets and baboon to study evolution of the eye. We've been working with collaborators, Mandeep Singh's lab, to transplant these. So far, we've been doing human into mouse, but we're now starting to move into using these non-human primate systems to go to intra-species transplants. So in other words, macaque to macaque, et cetera. And then the last big thing is we're branching out, kind of, again, getting this really big question, or how are all the cells of the human eye specified? And in particular, we're working on our retinal ganglion cells, the cells that connect the eye to the brain. And what you're seeing here is an axon outgrowth assay where we take a human retinal organoid, we put it down on Matrigel, and within 1 day, we can see extensions of axons of over 1 millimeter. So with that, I'll conclude. I'll thank everybody in my lab. This is when we were still virtual. I guess we need a new lab picture. I'd like to thank Kiara Eldred, who did the work on the first story. Kasha Hussey did the work on the second story, and Sarah Hadniak. And I'd like to thank you for your attention. I'll take any questions. Thank you again.
Thanks very much, Bob. That was a great talk. So I think we've got time for just a few quick questions before we go to the sort of early career session. So just see, we've got Matteo, just unmute your mic.
Yeah,
okay.
Hi again. I was wondering about this last point you made about the retinoic acid. So if it's not an intracellular choice between green and red, but it's a factor like retinoic acid, shouldn't we get in the eye sort of like concentric circles of how you have expression of green or red opsin, especially in the parts of the retina where it's very salt and pepper?
Sorry, can I ask you to repeat your question? And if you want to show your video, that's cool. If you don't want to, that's cool too. I was toying around with taking the sharing off, so I got distracted. So could you re-ask your question? I'm sorry.
Yeah, no worries, no worries. I was wondering about the red versus green. I always thought it's an intracellular decision, so each cell makes the stochastic decision. So it's interesting if instead is due to something like a factor like retinoic acid that I guess can come from outside the cell. I'm wondering whether, like, shouldn't we then have a pattern that doesn't look like what we see in the human eyes, but more like concentric circles, you know, more like a pebble in the pond?
Well, that's what we're working towards understanding. So basically what we think we're seeing is that in the central region, it's a mix of red and green cells. And then as you go out, it's only red. And so it's a bit complex, right? In that when you have waves, what we think is happening is in the first wave, you're getting blue, right? So if we started from the center and going out, we're making blue cones. And then the next wave would be green cones, but those green cones are generated in between the blue cones. And then the next wave would be red cones, and those are generated between the red cones. What we don't know, what we have is with adaptive optics, we haven't done these studies, others have, that the red and green, there's a lot of, I don't say controversy, but there's mixed opinions about whether that's a regular pattern or it's a random pattern. And so what I would say is this, I just want to emphasize that we definitely don't rule out stochastic mechanisms. And what it could be is that, think about if you had a gradient, right? And you had very high levels of the gradient, all the cells might go to one way. If you had very low levels, they would go the other way. But if it was somewhere in the middle, some cells might choose one side and one cell might choose another way. So in essence, it's a mix of both extrinsic and intrinsic variability. Does that make sense?
Yeah. I guess, yeah. If you phrase it as a mix of them, yeah, it does make sense.
Cool. Thank you for your question.
Thanks.
And we've got a question from Richard, if you just enable your mic.
Hey, Bob, how you doing?
Hey, what's up?
Sorry, I can't seem to turn my camera on. I don't know why. Bob, I just had a
face. Sorry, Richard and I go way back. So
I had a question, just something I noticed right at the start. So when you take the organoids out to 360 days, there's actually quite a big drop off in the red numbers of red greens, and I wondered if that was somehow physiological. You know, is that an important degeneration aspect or something, or is it just an artifact of the system?
Yeah, so we don't know. We think that it's probably that the optimal health of these organoids is about day 270. And so we think that a lot of them are dying off. We're starting to build the tools, unlike in worms, it takes a long, hey, how you doing? It takes a long time to build these reporters and stuff, but what we're trying to do is build kind of lineage tracing tools where we mark cells and then see what happens to them. And it's really hard to convince people to study those really late time points. So
yeah, of course.
That's kind of why we kind of picked day 200 as our like end point, because we knew like, okay, these are pretty well along and they're not even really the end point. What I will say is that when we play with the system, we see all kinds of dramatic effects. When we mess with that Dio3, we see a huge spike in the generation because we think we're getting more T3, we're getting a ton of cones, but then they actually decrease over time. And what we don't know is, are they dying? Are they converting into something else? We don't know.
Cool. Thanks, Bob.
Good seeing you, man.
Yeah, you too.
Great, thanks. So if we ask the postdocs and PhD students to stay online now, and we can have our sort of postdoc ECR session. So yeah, please feel free.
Everybody for coming.